the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Ketosis, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Gábor Somlyai, we're just working on the pronunciation. You're calling in all the way from Hungary. Thank you for joining me.
Thank you for having me.
Do you want to start off by just telling everyone a little bit about who you are and what your background is and what you research?
I graduated as a molecular biologist very, very long ago. I worked with DNA during my study and even after I graduated. But the reason I want to become a biologist because my aim was to do something on cancer research, hoping that I can find or figure out something which can be very important and useful for everybody. So I started that type of research with deuterium and deuterium-depleted water after I came back from the United States, where I was a postdoc position in a gene mapping work. This is what I did. But finally, I come back to Hungary and I looking for job, and I went to work to the National Institute of Oncology. So I started, because I had a PhD, I had the right to do research what I wanted. The aim of the research was to figure out whether this deuterium has any role in the living organism. And in the very first study at the Institute of Oncology, I could prove that a lower the concentration can cause very, very significant effect in tissue culture or mice had been transplanted with tumor. So since that time, our job is to generate more and more research, to involve more and more scientists, and to test on a different field of the biology the effect of deuterium in the living organism. Mm-hmm. So, and this is where we are now. I could say that the concept that the deuterium has a very important role in the living organism well established. This year we just organized the 5th International Congress on deuterium depletion. People coming from 13 countries, and we can see that searching on the PubMed for deuterium depleted water, the number of papers exponentially increasing since we published our first paper in 1993. Mm-hmm. So finally, my job is to started with a concept, with an idea that the deuterium has an important role. After that, started the research. We published paper, and now aim is to get a cancer drug registered for human consumption.
So we're going to be talking about deuterium and deuterium depleted water today. This is a new area for me. I don't know much about this at all. I have been hearing more about this, and I think people in general have started to hear more about this. But I think most people listening will know very, very little about this. So we have to start with the very basics today. So we're talking about deuterium, which is a version of hydrogen. So there are different types of hydrogen that differ based on their physical properties. Let's just start with the very bare bones basics here. What are the different types of hydrogen, and what is deuterium?
Okay. So in nature, there are three different hydrogen. The hydrogen with mass number 1, deuterium with mass number 2, and the tritium with mass number 3. That this is a radioactive. The deuterium is a non-radioactive version of the hydrogen. But in that case, there are 100% difference in the mass between the hydrogen and deuterium. And that causes very big differences in the chemical reaction when we talk about hydrogen or deuterium. Typically, an oxygen H bond split 10 faster than an oxygen deuterium bond. So it is called isotopic effect. This is well known since the discovery of the heavy hydrogen, the deuterium. The reason the deuterium can be important, that even everybody knows that deuterium is present, but in a surface water, in a normal water, the ratio between hydrogen and deuterium is about 6,600 to 1. So we can say that very rare, the deuterium. But if we explain the deconcentration in millimole, because we know that in a calcium level in the blood about 2.5 millimole, the kalium, potassium is about 3.55 millimole, but the deuterium is 12 millimole or 13 millimole in our body. And considering that here is the deuterium in a so relevant concentration, and if we see that calcium, magnesium, potassium, sodium is so strictly regulated in the living organism, but the deuterium 10 times higher than the magnesium, and nobody investigated the possible role of the deuterium. So the breakthrough in my way of thinking and the research, that address the question, what will happen if we reduce the deuterium level and whether it causes any effect? Because if we see some significant effect, we can suppose that during the evolution, the evolution gave some role of the deuterium. Mm-hmm. So that was the question.
Yeah. So obviously, our bodies are made out of, there's lots of hydrogen in our bodies. There's lots of water, and water is H2O. So 2 hydrogens, 1 oxygen. There's 3 types of hydrogen that we know about in the universe. There's hydrogen, what we call, let's just call it regular hydrogen. And that's basically just 1 proton in the nucleus. So then deuterium would be a proton and a neutron. So that nucleus is twice as massive in the deuterium compared to the regular hydrogen. Both of those are non-radioactive. And then there's tritium, which is 1 proton, 2 neutrons. So it's got 3 times the mass of the nucleus of regular hydrogen, which is that single proton. That one is radioactive. Today we're talking about regular hydrogen compared to deuterium. And basically what you said is, so the deuterium is way more rare than regular hydrogen if we just measure it out in the world and measure it in our bodies. But it's still more plentiful than some other things in our bodies that we know are important.
Yeah. Exactly. Yeah.
So when we think about this just in raw physical terms, so the deuterium's twice as massive as the regular hydrogen. So if we imagine a water molecule, most water molecules in our body and in the world are 1 oxygen and 2 regular hydrogens, but sometimes they can have an oxygen with 1 or 2 deuteriums. So they're going to be heavy. It's going to be a heavier, more massive water molecule.
Typically, at that low concentration, the water is in HDO form. So one hydrogen, one deuterium, and one oxygen. It's very rare at a very low level of deconcentration that two deuterium bind to the oxygen. So if you talk about heavy water, in that case, the deconcentration is close to 99%. Yes, at that case, two deuterium bind to the oxygen, but at a natural concentration, we can talk about HDO. This is the way how the deuterium is present in a water form.
Yeah. And I guess if we think about this, so there's water, and most of the water is going to be normal, regular water, how we would normally think about it. H2O, both of those hydrogens are the regular hydrogen. Sometimes you've got a deuterated heavy water, and that's H2O, but one of the hydrogens is this deuterium hydrogen, so it's a little bit heavier. So I guess it's literally going to be heavier, and that would presumably affect the water in terms of just like the physics of how it moves because its mass is different.
I would say yes, but the point is that in our body, one single cells, there are billions and billions chemical reaction every second. And the hydrogen and the hydrogen bond has a very, very strict role in the DNA, within the DNA, when the messenger RNA is synthesized, then the base pairing between the DNA and the RNA, when the ribosome is synthesizing the proteins. So it's very crucial. So billions and billions reaction are running with the hydrogen, splitting the hydrogen bond and forming a new hydrogen bond. And the question is how frequently that reaction is running with the deuterium, which slows down that process, maybe stop the whole reaction, slow it down, and what is the effect for the next step of that chemical reaction, and how can it modify that whole process?
Yeah.
So the reason we should think about when I talk about those who are not really expert, we should understand how the life can be organized and how can be so sophisticated, so perfect. People should think about that there is a cell size which is, let's say, 40 micrometer, which is 25th part of 1 millimeter. And there is a nucleus which is 10 micrometer. Can you imagine how small is that? But within that nucleus, there is 1.8 meter length DNA. Everybody has the DNA packaged in the 10 micrometer size nucleus with a 3,200,000,000 base pair. And it's a so condensed information containing the story of the evolution, containing all the knowledge, everything about the people, the color of the hair, color of the eye, everything. And it is packaged. And the question is whether the life can be organized by big protein molecules. What we suppose, and we are building the drug development, that finding a mutation in the gene and hoping that if we can target that protein, that we can get a cancer drug. And this is how the world is try to solve the cancer problem. But when we talk about that, how the life can be organized and how the cell regulate the cell division, then I say, based upon our research, it is organized on a submolecular level. So during the billions of years, when finally the living organisms could use out that there is a heavy hydrogen, they built up a regulatory system. Changing the cell, they are able to modify the DH ratio in the cell. And after that, changing DH ratio simultaneously can send the message to all the molecules, all the proteins, all the nucleic acids within the cell. So this is a change of paradigm. So far, we believe that finding a molecule binding to one protein, a kinase or whatever, that can be so effective that we really can intervene and stop the cell growth and cure the cancer. But the aim is, how can we control the whole system at the same time? And this is what we are saying. So if we talk about only the prokaryotes, the bacteria don't need the deuterium because they are so primitive organisms that they can organize the cells metabolism based on the molecular level. But when the mitochondria, which was a bacteria, targeted and moved into the eukaryotic cell, and they live together, that give a chance for the living organism to set up a new system. And we say today that the membrane transport can increase the deuterium hydrogen ratio. Meantime, everybody, every day, by the mitochondria, we synthesizing water. So we effectively produce every day about 0.3, 0.4 liter metabolic water. So we are burning the hydrogen to water. This is a metabolic water. And that way the mitochondria producing deuterium-depleted metabolic water. So, and this is a mechanism how it works, the heavy hydrogen, the semi-heavy hydrogen.
So, okay, let me see if I'm following so far. So you've got deuterium and you've got regular hydrogen. So we can abbreviate those to H for regular hydrogen, D for deuterium, which is twice as massive. There is some water that is heavy water because it's got a deuterium hydrogen as part of the H2O molecule. And the presence of a regular hydrogen or reverses a deuterium hydrogen in a water molecule will change how easy it is for the bond to be broken between the hydrogen and the oxygen. And I guess what people should understand going into this is, a big reason why water is essential for life is not only because it's a solvent that we're immersed in, but it's participating in biochemical reactions. So what you're saying is billions of times per second in all of our cells, we're constantly breaking the hydrogen-oxygen bonds of water as part of the biochemistry of life. And so if the strength of the bond between a hydrogen-oxygen and a deuterium-oxygen is different, then it would have to be the case that the concentration of heavy deuterium-containing water would affect things like the rate of all of the chemical reactions in a cell that depend on water.
So there is one more comment to you. Not the water is the only molecules in our body which has hydrogen and deuterium.
Yeah.
All the molecules, all the amino acids, all the nucleic acids has the hydrogen and deuterium. So when we are talking about deuterium depleted water, this is not the reason that. So the key is that using deuterium depleted water, we are able to modify the DH ratio in all the molecules where there are the hydrogen in an exchangeable position. So when and not chemical reactions running only with the water, but there are 2,000 different chemical reactions in one single cell. Typically, most of the reaction has a hydrogen or deuterium. So finally, when we talk about hydrogen deuterium, we talk about all the molecules, all the chemical agents, all the amino acids, everything which has either hydrogen or deuterium in a given bond or in a given position during the chemical reaction.
Yeah. So if deuterium is present in a hydrogen, and that heavy hydrogen is bound to something, it takes more energy to break that bond. Is that what we're saying?
Yeah.
So that should mean it should slow down the rate of reactions. Yes.
Yeah, those chemical reaction which is running with heavy hydrogen, it's much slower.
And that's been measured. Have people measured that directly, and we know that for sure?
Yeah, sure. Yeah. There are. It's a long story that everybody knows that which chemical bond, how strong and how slow, depending on the hydrogen deuterium. But in our case, let me tell you one experiment which was published. This is related to diabetes. Okay, one day a patient with cancer and with consuming DDW, this is a deuterium-depleted water, and came back and he said that his blood sugar level went down because he was a diabetic, not only cancer patient. So, and that, I went to the Semmelweis University and we ran several rat study. So we made the rats diabetic, and the question was whether the drinking water of the deuterium can reduce the blood sugar level of the rat. At the first time we used 25 ppm and 150 as a normal. So the normal deconcentration is 150 ppm, which means there are 150 deuterium out of 1 million hydrogen. So, and we found that when the rats received insulin, then the 25 ppm reduced the blood sugar level of the rat, which is very good, because we can say that with a lower deconcentration, we could reduce the blood sugar level of a diabetic rat. But then we ran other study with 75, 105, 125, and we went up to 150 ppm. And it turned out that the best deuterium level is between 125 ppm and 140. So not the lowest deconcentration reduced the best way the blood sugar level of the diabetic rat. So, and this is the reason I say, so there is a very specific signal system in the cell. They recognized that for the insulin signal to send the message to the cell, the 125 and 40 ppm is the best to reduce the blood sugar level. And I don't know, do you know the meaning of GLUT4?
I do, yeah, but I think we should explain that for people, yeah.
Okay. Yeah, so a GLUT4 is a protein which is responsible to pick up the glucose from the blood. So then we checked whether the deconcentration can modify the GLUT4 concentration in the membrane of the muscle. And when we checked it, it turned out that the GLUT4 concentration was the highest in the membrane at 125 ppm.
So you're saying you changed the deuterium levels in the water and across different concentrations, and changing the amount of deuterated water in this system changed the amount or the activity of a protein that's responsible for regulating glucose levels.
Yes. Yeah. So when the insulin binds to the membrane and send the message by the receptor, they stimulate the GLUT4 translocation from the cytosol to the membrane. And based upon that result, it's obvious that the insulin signal system works much better within 125, 140 ppm range than at 150 ppm. And then we ran a human study, the people consuming 105 ppm for 90 days. And at the end of the third month, we could reduce the fasting glucose level, and at 30% of the population, we reduced the insulin resistance. So, and the reason it's very important, because there was a big change in the diet at the 70s, when in the United States, it was said, change the calorie intake from lipid to carbohydrate. So when we are eating carbohydrate, our mitochondria will synthesize metabolic water with 150 ppm. So the today's population average D level is much higher than it was 60, 70 years ago, when the people were eating much more fat. Because from the fat, when we burned the fat in the mitochondria, we produce 110 ppm metabolic water. And this is a big game. So there are 100 or 400 diabetic patients in the world, because the average deconcentration is close to 150 ppm.
So,
and the insulin signal system is not so good for that.
yeah, I want to get to carbohydrate versus fat metabolism after we talk about mitochondria, after we talk about some more basic stuff. But let's back up even further. So you've said things like 150 ppms, 140 ppms. Let's start with, what is the deuterated water and the deuterium concentration of water out in the world? Like if I go scoop up some water from the lake or from the ocean, what is the concentration of this deuterated water?
In the ocean, in the Indian Ocean, it's 155 ppm. And this is a standard. When the clouds appear in the equatorial area and the clouds are moving to the north and south pole, we could say that the heavy hydrogen, semi-hydrogen, fall out earlier. And let's say I live in Europe, Middle Europe, here it's about 145 ppm, the deconcentration. If in Norway or at the North Pole, it should be about 130 ppm. Even when we going up to the hillside, increasing the altitude, that way the deuterium concentration again decreasing. So there is, let's say, 20 ppm fluctuation or changing depending on where we are on the Earth.
I see. So around the equator, basically in the ocean, you would find it deuterium water at about 150 ppm. So that means 150 molecules of water have a deuterated hydrogen out of 1 million molecules of water?
Yeah. Yeah.
And then because deuterium is more massive than regular hydrogen, as you go up in altitude or as you go towards the poles away from the equator, water is evaporating, water is falling, it's moving around like water does, just as a consequence of the physics of all of this. Because deuterium is more massive, the water closer to the poles is more deuterium depleted. The deuterium is sort of falling away, and there's less deuterium, there's less concentration near the poles compared to the equator.
Yeah, that's the question of the evaporation. So because the semi-hydrogen, semi-heavy water is bigger mass, stronger bond, it means the boiling point is higher. So when you are boiling the water, the H2 molecule much easier can go to the atmosphere than the heavier semi-heavy water. So this is how we can separate the heavy water, the deuterium depleted. This is how we produce deuterium depleted water. We are producing tons of steam to getting a couple of liters of deuterium depleted water during that so-called fractional distillation.
Got it. So that's how people make deuterium depleted water that has less deuterium than the water surrounding it. You're basically just doing a lot of boiling, a lot of steam generation, and then you're separating out the water from the steam. And because the regular hydrogen containing water will evaporate more easily, you can fractionate the water. You can separate it and get deuterium depleted water with less deuterium in it.
Yeah, that's it.
Okay, so 150 ppms roughly around the equator. That will change as you go to different parts of the Earth. So as you go north away from the equator, the ppms, the concentration of deuterium in the water, will tend to go down. In a normal living, breathing animal, a normal living, breathing person, how much deuterium do our cells and tissues tend to have compared to the external water in the environment?
So typically we say that people should drink 1.52 liter water per day. And the other water what we gain is coming from the food, and not the food which water is in the food when the hydrogen is bounded to the carbon source, glucose, amino acids, or lipids. So when we're talking about metabolic water, in that case, the hydrogen is coming from the glucose hydrogen, from the lipid hydrogen. And that, in that case, there is big difference in the DH ratio.
Yeah. So let's define this now clearly for people. You said metabolic water. Describe that in very basic terms for us. How does our body make what we call metabolic water? What is that?
The metabolic water is made by the mitochondria. So the mitochondria can produce the ATP that within the mitochondria, from the inner part, the hydrogen is transferred to between the two layers, and based upon the hydrogen concentration coming within the mitochondria, and that type generating the ATPase, which can finally synthesize the ATP. And the energy, which was finally based on the sunlight, the plant gained the energy from the sunlight. That way they make from carbon dioxide glucose. So from carbon dioxide and water make the glucose. And when we oxidize that glucose, it's going back to carbon dioxide and metabolic water. So that is the circle of on the Earth. And that energy which is coming from the sun, captured by the plant, we get it back when that hydrogen flows through the ATPase, generate the energy, building the ATP, and that ATP is used up in the cell.
Yeah. So basically, right, our cells are filled with mitochondria. This is the site where most of our ATP, the energy currency of the cell, is produced. It's produced because the mitochondria have two membranes. They're creating a proton or a hydrogen gradient, and those hydrogen ions, those protons, are flowing through the ATPase enzyme, which is basically a little molecular rotary motor. And so the flow of hydrogen through that little protein spins that motor, and we're making ATP, but we're also making water molecules. And that would be like why our breath contains water vapor when we exhale.
Yeah, we need breath, because when the hydrogen is coming, need the oxygen, and that they get the electrons, and then we finally can synthesize the metabolic water.
Yeah. And what's powering all of that is the proton gradient, but also we're oxidizing or burning our food, our carbohydrates or our fats. And that's feeding into this process to help create that gradient and make the ATPase do what it does to eventually create this water. So, what else is going on in the mitochondria here that's relevant? So this might be where we start to talk about where this deuterium-depleted water comes from and where it is within the mitochondria.
So the TCA cycle is running in the mitochondria. So this is the basis that finally generate the free hydrogen, and that's what you explained so properly. But based on the regulatory system, what we say exists, the big game is how the TCA cycle is running, what is what is the food we are feeding the mitochondria, so whether it's carbohydrate, lipids, or whatever, and that will determine the final deconcentration of the metabolic water. And the key issue is everybody knows that there is one common feature of each cancer cells, and that is that the damaged mitochondria. So if we say there are healthy cells and cancer cells, 90% of the cancer, the mitochondria is damaged. So we can say no metabolic water production in the cancer cells. It means no deuterium-depleted water production in the cancer cells. And that is very important because on the other side, when a growth hormone binds to the receptor and stimulate the sodium hydrogen antiport, so in that case, the cell pick up one sodium and transport one hydrogen out. We published more than 30 years ago that process will refer to eliminate the hydrogen. So when the growth hormone stimulates the sodium hydrogen antiport, that will cause an increase in the DH ratio. And when we talk about the key regulatory system, the question is whether a cell are able to lift up the DH ratio to trigger the cell division. If you have a healthy mitochondria producing deuterium-depleted water, then it's very difficult because the mitochondria is producing the deuterium-depleted metabolic water, so it's counteract with the increasing membrane transport. But in a cancer cells, the cells very easily lift up the DH ratio, and this is the reason that finally the people can get a cancer. So that is a key mechanism we say very important.
Mm-hmm. So I wanna really connect the dots here between the deuterium depleted water and the metabolic water. So water is being produced as a consequence of mitochondrial metabolism. I get that. The hydrogens are flowing through the ATPase. We're oxidizing our food, our fats or carbohydrates, and we're producing water in the mitochondria as a consequence of that oxidative metabolism. Why is the water produced, this metabolic water produced inside the mitochondria, why is it deuterium depleted? Why does it contain less deuterium? Where is the deuterium leaving the equation here?
Good question. Yeah. So just because we talk about that, the D bond is always stronger than the H bond, it means that in each chemical reaction, always the hydrogen will be preferred. So at that cause when the plant synthesizing the carbohydrate, we could say that there are 6 carbon in a glucose. It's very easy. But when the cells synthesizing lipids, which means they taking the glucose source and making lipids, 2 carbon, 2 carbon, 2 carbon binding.
There's more
to each
steps.
other. Yeah, there are 16, 18, whatever, 24 carbon in one lipid molecules. That means in that process, as I mentioned, always the hydrogen will be preferred. So finally, here will be a big depletion for the deuterium. This is one thing, but there are more important things that in the plants there are 3 different pathways to fix the carbon dioxide from the air. There are a so-called C3, C4, and the CAMV. And based upon those plants, for example, the corn belongs to the C4 pathway. That is not so depleted than the wheat, which has a C3 pathway. So even depending on what type of plants you eat,
right,
that can cause differences in the deconcentration.
I see. So literally what you're saying is, so let me translate 2 things here. So there's different types of plants, just like there's different types of animals. C3 and C4 plants are plants with different ways of doing photosynthesis. So the biochemistry is just happening in a different way. The level of deuterium in C3 compared to C4 plants is different. And the reason the level of deuterium is going to depend on C3 versus C4 in a plant or carbohydrate versus fat metabolism in our own cells has to do with the number of chemical reactions it takes to do stuff. Because the deuterium is heavier and the carbon-hydrogen bond is stronger and therefore more difficult to break, then the more chemical steps, more chemical reactions you have in a system, each time the hydrogen is going to be preferred to the deuterium. So statistically speaking, when you have many more reactions that need to happen to, say, do lipid metabolism compared to carbohydrate metabolism, you're going to have more of that deuterium falling away, and therefore you'll produce more deuterium depleted water.
Yeah, exactly. Yeah. Do you know the capsaicin?
The
Capsaicin, which makes the pepper
spicy?
hot.
Yeah, capsaicin.
Yeah. Do you know, it's a very, very complicated molecule. The D level is about 60 ppm. So just because synthesizing one capsaicin molecule is so different, so much biochemical step, so it brings down the concentration of that specific molecule.
Yeah. So to summarize here again, just to repeat this, because it might be a little bit hard for people to keep track of all of this if they're new to it. So there are more chemical steps involved in breaking down lipids than carbohydrates. And so that means, for each of those steps, you're going to prefer the hydrogen to the deuterium, prefer the hydrogen to the deuterium, and so on and so forth. So fat oxidation, lipid oxidation, produces more deuterium-depleted water, less deuterium in the water compared to carbohydrate oxidation. Similarly, and for some similar physical reasons, different types of plants that do photosynthesis in different ways and involve different biochemistry, they have different levels of deuterium inside of them. And therefore, eating wheat versus corn, as you said, is going to put a different amount of deuterated hydrogen in your own body.
Yeah. The pineapple that, for example, can enrich the deuterium.
I see.
So when somebody eating pineapple, that should be even over 150 ppm, because this is the third type of chemical reaction, chemical way to fix the carbon dioxide.
Yeah. Okay, so deuterium's out there. It's in the environment to some extent. Its concentration depends on where you are in the globe, and its concentration depends on the type of biochemistry an animal's doing, the type of photosynthesis a plant is doing, like C3 versus C4, the type of food that you're eating, like carbohydrates versus lipids. And then at different locations in the cell, like in the mitochondria, you can get different levels of deuterium depletion based on how the biochemistry works. So there's different concentrations of deuterated water in different parts of the world and in different parts of our body and in different organisms. What do we really know about the effects of different deuterium concentrations, different concentrations of deuterium depleted water on biology? And let's start here with the basics. Let's start with in vitro experiments. When you're studying cells in culture in a petri dish where you can directly control the deuterium depleted water levels, what have you observed and what have others observed about how deuterium levels affect basic cell processes like gene expression, cell division, and that type of thing?
Oh, that's a good question. Yeah. So the very first study, of course, I did the simplest way. I prepared media for tissue culture with deuterium depleted water. And the very first paper we published, I used 25 ppm, 150, and I even tested the 300 and 600 ppm. Yeah. So the deuterium enriched water also.
So again, just for reference, 150 ppm, we might call that sort of the normal background level.
Yeah.
So lower than 150 is deuterium depleted, and then higher would be deuterium enriched.
Yeah.
Okay.
So and what we found that reducing the D level below 150 ppm, that way we could slow down the cell growth, but increasing over 150 ppm, that stimulated the cell growth. So which means that the higher D level, close to the 150 ppm, it was very advantageous for the cell to start the cell division. So that was the very first. Later, there is a very extremely good technology recently. In that case, the cells, we tested again, the cells were growing on a gold layer. So in that case, we didn't need to manipulate with the cell. We just followed how the cells can modify the impedance of that gold layer. So it makes us very precise following what the deuterium can do.
So hold on, you're saying there's literally cells growing on a layer of gold, and it's changing, the impedance can change. So you can kind of measure how well electrical currents are being conducted.
Yeah, the faster they grow, the faster they modify the impedance of that gold layer. So in that case, we could prove changing the D level from whatever over and below 150 on dose dependent way, reduce the growth rate. At 40 ppm, we completely could stop the cell growth. Over 150 ppm, we stimulated. But in another study, we investigated how sensitive for the deconcentration. In that case, we run a study that every 8 hours, we change the deconcentration with 1 ppm. And we found that reducing with 1 ppm every 8 hours, that resulted in a slower growth rate. So the cells are so sensitive and so precise and measuring the D level that 1 ppm decrease resulted a slower growth rate. So, and that is very important to see that how precise system we are talking about.
Yeah. So basically, the concentration of deuterium in your water, you just have cells growing in a petri dish, and you're giving deuterium, or you're giving water with different levels of deuterium in it. When the deuterium
Yeah,
levels are lower, under 150, that slows down growth. Cells grow
yeah,
slower the less deuterated water is in the medium.
yeah. The more lower the D level, the more slows down or completely stop the cell growth.
Yeah. So if you increase or you enrich the deuterium levels in the water, cell growth speeds up. That's a little bit, I guess, counterintuitive to me, as someone who doesn't really know about this stuff, because if there's more deuterium, there's more of that heavy water, that bond should be stronger. I would intuitively think that that would slow things down.
Yeah, you are right. But it depends on. So when in the very first study, when we goes up to 900 ppm, that slows down. But within the range, 150 ppm plus minus 30 or 30 ppm, the question is not the physical differences between hydrogen and deuterium, but this is a sensitive system for the DH ratio, where a very small changes in DH ratio are amplified and enhanced by the enzymes by the cells.
I see.
Because small changes, for example, there is a protein which is in a given conformation. If we change the DH ratio, that less or more deuterium can fix the conformation of the enzyme, which will be maybe more active or maybe inactive. So small changes in the DH ratio, an enzyme could be even faster
I
or
see.
slower, and that increase the effect of the
Yeah.
deconcentration.
I think if I'm hearing you correctly, what I'm hearing is if you go across a very wide range, you can see differences that are different than if you're in a narrower range. So a cell on Earth will never see 0 ppms, and it will
Yeah.
never ever see 900 ppms. But
No.
within the range that biology actually encounters out there, 150 ppms and a bit lower, a bit higher, you're saying in that range is sort of where the action is. It's where cells
Yeah.
have evolved and therefore what they're sensitive to.
Yeah. Yeah. This is exactly.
Interesting. So, what else have you seen in vitro experiments other than cell growth? What other things have been looked at that are sensitive to the deuterium depletion levels of the water?
So our recent paper, we have published a couple of papers, what will happen with the genes, because this is one of the key issue. Uh, we know hundreds of genes having key role in the cell growth, and the drug development is based on finding the mutated genes, and based upon they try to find the new drug. So the question is, what will happen with the gene expression when we modify the deuterium hydrogen ratio? And in that study, we used 40, 80 ppm as a deuterium depleted water, 150 and 300 ppm. And we could check over 500 kinase gene and over 200 cancer-related genes. And the point was that 97% of the genes responded for the higher deconcentration. And why can be important? Because, as I mentioned, when a growth hormone binds to the receptor, stimulate the sodium hydrogen, that will cause an increase in the DH ratio. And if the DH ratio is increasing, that can simultaneously activate hundreds of genes, which has an important role to manage the cell growth. And this is a big difference between the cancer drug development and applying DDW in the cancer therapy, because the cancer drug will target one gene, one mutated genes. By modifying the DH ratio, that way we will not allow all the 100 cells to be activated, because we keep the D level at a much lower level. So the cells are, even the sodium hydrogen transport is activated from the low D level, they are not able to lift up the DH ratio to the threshold. So that is the reason that we not targeting only one signal pathway, but using and people drinking DW, they simultaneously can impact the whole network of this cell growth regulation.
Yeah. Well, I think, I think one of the points I think you're trying to make is I'm already starting to change how I'm thinking a little bit in the sense of in what we might call normal biology or normal drug development, we think about one protein binding to one receptor or one drug binding to one receptor, and then this pathway turns on and then it goes to the gene. But what you're talking about is basically the weight and the viscosity of all of the water can change the speed and the patterns of all of the reactions everywhere all at once. This is more of a distributed biophysical thing, not a one gene, one molecule, one protein pathway way of thinking about biology.
Yeah, that's it.
And just
this is what I try to explain.
Yeah, let me see if this analogy works a little bit. I'm trying to play with this in my own mind right now. So we talked about the deuterium concentrations in the water, and we said the more deuterium depleted you are, the less deuterium, the more it slowed down growth relative to 150. And then if you increase it, it sped it up. But then you said, if you really increase it a lot, it will in fact slow things down. And I guess the analogy I'm thinking of here is like, I could go swimming in a pool and it could be freshwater or salt water. And they'll have different levels of salt in the water, but I can still swim through it. And maybe I can kind of tell there's a little bit of a difference, so it affects my behavior. But if I go try and swim through molasses or tar, it's just too big and I can't swim. But I'm also never going to go swim inside of molasses for that reason. So, is this kind of maybe how to start thinking about it? We're talking about the viscosity or the weight of the water.
So there is a limit, and I suppose that for different cell lines there are a different part of the limit. So within a range for, I don't know what type of cell, we can increase up to 300, 400, but another cell line, maybe we can even higher, and we do not see that flashback that the deuterium finally even inhibit. So the point is, going back to the insulin, that there are receptors and there are indicators for this DH ratio, which can multiply very simple changes in the DH ratio, and then they can multiply 10-fold, 100-fold.
Yeah. And can you give me an example of that? How is this DH ratio actually being read out by certain proteins?
What do you mean?
So describe how this amplification process is happening. Are there actual literal sensors in the cell, like certain proteins that are directly sensing the DH ratio?
I suppose that each of the proteins recognize the changes in the DH ratio and must be very, very precise and key players of that whole protein system, which are more sensitive and more important role they have, how they react for a changing DH ratio. So based on the gene expression study, we could see that there was a couple of genes we can get over 100% increase in the gene expression, and there was a 50% increase. So it depends on the genes, for example, for example, those mTOR gene, we could get a 100% increase in the expression at 300 ppm. We know that the mTOR is a very important key player in that signal system.
Right. So mTOR would be a key protein information pathway involved in growth. It's often dysregulated in cancer. And you're saying increasing deuterium levels in the water increases the expression of mTOR in certain cell cultures.
Yeah. The other point should be, do you know the Herceptin?
No.
The Herceptin is a very good cancer drug. At that cover the receptor. So when the growth hormone binds to the membrane, it cannot bind to the receptor. And it means that the cell will not stimulate the sodium hydrogen antiport. So they are not able to lift up the DH ratio. And what we see that those patients who are receiving the Herceptin plus consuming deuterium-depleted water, we've got a much better data, it's much better results than only receiving the Herceptin, because we are targeting the same system. With deuterium-depleted water, we bring down the deconcentration, so that way we make it very difficult for the cell to increase, but at the same time the Herceptin not allow for the cell to stimulate the sodium hydrogen, and that way try to increase the DH ratio. So, and there are other kinases, and we saw that giving the drug, combining with DDW, we can get a much better results. So sometimes patients receiving the drug, no real good response. Combining with DW, that way we can increase the efficacy of the drug. Which means somehow in that case we targeting that given protein which is targeted by the drug.
Okay, so you can change the deuterium levels in water, put cells in a dish growing in this water with different concentrations of deuterium, and you see effects on cell growth rates, how often the cells are dividing. You can speed it up, you can slow it down when you play within a certain range of deuterium-depleted water. You can affect gene expression up or down, depending on the concentration, depending on the gene. All of that cells growing in a dish. Let's now talk about animals. When we do experiments in animals like lab rats or mice or something like that, has anyone done experiments where they're supplying deuterium-depleted water, increasing or decreasing the deuterium levels, and looking at cancer rates and anything like that?
The first paper, what we published in 1993, we mentioned a mice study. So in that case, we transplanted human breast cancer to the mice. And one group of the mice consumed 150 ppm, the other 30 ppm. 60% of the animals were still alive and they were healthy. On the control group, only one mice survived somehow the tumor. So it was evident that when the mice consumed deuterium-depleted water, that way we could stop the tumor growth in the mice, and finally the tumor disappeared, 60% of the mice. And after that we studied other mice study, but the point is we've got a registered drug for veterinary use. So we tested on dogs and cats when we replaced the drinking water with 25 ppm DW. In that case, we say roughly 50% of the animals we can get a complete response, the tumor disappear, and the other 20-30% we can get a partial response. So that is on the market, and the pets owner like it, buy it, use it, and sending the pictures about the dogs on the Facebook and showing that how happy they are that the animals are fine.
Okay, hold on. So in the rodent studies, you're basically grafting human breast cancer tissue into a rodent, and then you're saying we're going to give them regular water in one group, and then we're going to give them deuterium-depleted water, lower deuterium in the water for the other group. And you said the deuterium-depleted water group saw slower cancer growth and or lower cancer rates?
I would say in that study that we published in the first case, we eliminated the tumor from the mice.
So the tumor went away?
Yeah. I wouldn't say all, 60% of the mice 60%. Survived.
Yeah.
Yeah.
And what was the control group? What did the rate of death look like?
One might survive out of 11 in the control group. So I would say in that mice, I suppose that the tumor couldn't start. Otherwise, all of them would have died.
Okay, and then you said, okay, you've also got a veterinary drug that's given to cats and dogs when they have cancer, and you're saying when you give deuterium-depleted water to cats and dogs with that drug, they see better outcomes in terms of cancer?
Yeah, yeah.
And has that been published?
So the drug itself, yeah, we published mainly in Hungarian about, I have a couple of English paper published with dogs showing the size of the primary tumor and the size was decreased. We published, I guess, 2 or 3 papers. We have a paper ready for publication with the animal study, but that latest one has not been published.
Yeah. Do you, little side note here, do you publish papers yourself and just the scientific community in Hungary? Are there papers published that are not published in English, that are only in Hungarian?
No, I would say all the paper was published in English, only a couple was published in Hungarian.
Okay.
Yeah. So, and then, as I said, on the PubMed, there are about over 200 paper appear searching for DW. 100 is relating to the cancer and all this stuff. Most of them is not published me or our group.
Yeah. I want to back up a little bit and ask you something about more basic biology before we move on to some of the human studies you've been involved in. So we talked about mitochondria. Mitochondria are where a lot of the action is in terms of bioenergetics in our cells. We've got that proton gradient that the mitochondria set up. The hydrogen ions flow through the ATPase, the little motor that ends up creating ATP and also metabolic water. I would imagine that when I think about this biophysically, hydrogen ions are lighter. They can flow through at a different weight or maybe with different ease compared to deuterium because it's going to be heavier. Is that the case? Do we know that if you compare the inner mitochondrial space and the mitochondrial matrix, and you look at the flow of hydrogen through the ATPase, does the DH ratio affect the actual ATPase in terms of how fast it's spinning or its efficiency in generating ATP?
That's again a very good question. So I would like to see, and I try to run study just directly with mitochondria. So far I couldn't. But what we have done, we tested the effect of DW on athletes. So we made 2 groups of athletes and we made a loading test and we took the blood samples and then one group consumed DW for 44 days. And then we made another loading test, took the blood samples. And then we found that after consuming DW, the lactic acid appeared much later during the loading test. The anion gap was smaller and they regenerated much better. The glucose mobilization was better.
What was the loading test exactly?
They were rowing.
Oh, I see.
And 5 minutes break, 5 minutes break, 5 minute break. So 4 times we repeated, and at each time we took the blood samples. So based upon that experiment, it's in a race, it's critical whether the acidification, how it's running, and whether they can reach the 200 meters in a swimming pool. So this is what we found, that as the lactic acid appeared later, it means that the mitochondria could work properly and they could synthesize they had enough oxygen, they could synthesize the metabolic water, and they didn't run out of the oxygen. And the lactic acid, this is the reason it appeared later. Because when no enough oxygen and the mitochondria is not able to cope with that increasing hydrogen ion concentration, then it goes to the lactic acid, so they are not able to burn the glucose to carbon dioxide and metabolic water. So this is what we had. And I know that there are papers and talking about that, and it's very probably possible that when the Ds reaching the ATPase, that stay maybe longer in the position before flowing through on the membrane. It's very probably that way we can interact and modified effect of the ATPase.
Okay, but the basic result from the experiment you described is you got two groups of people and they're rowing. They're coming into the lab and they're rowing. And obviously, when you start rowing, you're going to start to sweat. You'll eventually get tired. Eventually, your muscles will burn. All of the normal things will happen when you do that type of exercise. One group was just drinking regular water, and one group was drinking deuterium-depleted water, lower levels of deuterium for a number of many days leading up to the test. The group that drank the deuterium-depleted water was able to basically row longer before lactic acid built up, and that would imply that they probably could have rowed further or stronger.
Yeah.
Interesting. And okay, so you've done experiments in people. You've seen effects in cell culture and in animals with, giving different levels of deuterium in the water. What studies have been done in human beings in a more clinical setting? Obviously, one of the things that people talk about when they get excited about deuterium and deuterium-depleted water is cancer. You mentioned the animal work already. Lower deuterium levels, meaning deuterium-depleted water, seem to go with lower rates of cancer or better outcomes in cancer in animals. Has any work been done with DDW in humans, human cancer patients, I mean?
So we could run one phase 2 double-blind randomized clinical trial with prostate cancer patients. So the reason I choose the prostate, because one of the first person had an inoperable prostate cancer. He started drinking DW, and a month later, they couldn't palpate the tumor, and there was a decrease in the PSA. So this is the reason I choose the prostate cancer. So in that was very, very long ago. Even I didn't know a lot of things about deuterium depletion. But anyway, that was a double-blind randomized clinical trial. There were bottles with a number. 5 numbers means normal water. 5 numbers was the 85 ppm. And neither the MDs nor the patients know what number means the DW. And what we found that there was 80% decrease in the PSA level in the treated arm, and there was only 47% decrease in the PSA level on the placebo arm.
And what is that? What is PSA?
The PSA, prostate-specific antigen.
I see. Okay.
That PSA is correlate with the size of the tumor. So we had 22 treated, 22 placebo person, and the cumulative number of the PSA was at the beginning, I don't know, about 800, and 4 months later it was 60% less, whatever. And so there was a big sharp decrease in the PSA volume, but more important was that every month the prostate size was followed by ultrasound, rectal ultrasound. And after 2 months, the MDs know that that number means the DW, that number means the placebo, because it was so obvious. There was 160 cube centimeter decrease in the prostate volume, and that was only 53 cube centimeter decrease in the placebo arm. So
I see.
3 times more.
You're basically saying you did an experiment, you gave 2 different groups of people with prostate cancer water. One of them got regular water, one of them got deuterium-depleted water. After a couple months, and I'm guessing they were getting some other treatment here, you're basically saying the physicians are used to seeing a certain rate of decrease in the tumor size, and it was so much bigger in the DDW group that they could actually guess which group was the placebo and which group was the treatment group.
Everybody received the conventional therapy plus placebo or DW. But the most important thing was that the study ran only for 4 months, and then we checked the survival after a year. So we lost 2 people out of 22 in the treated arm, but 9 in the placebo arm. So that was a phase 2 double-blind. And if we see that was a prospective study, and if we check all the retrospective study what we have done and we have published, this roughly we can say the survival is typically 3 to 7-fold longer if the patients consuming DW beside the conventional therapy comparing to those who only receiving the conventional therapy.
So basically, when you've given people deuterium depleted water, in addition to whatever treatment they're getting, basically it's like an adjuvant therapy. It's something you add to another treatment. You've generally seen better survival. So they last longer before dying, and or the tumors shrink faster in the DDW group.
Yeah, exactly.
Wow.
So we have published with lung cancer, breast cancer, prostate cancer, glioblastoma, pancreatic cancer data. That should be about 1,000 patients. But just a year ago, we evaluated all the patients we had. In that case, we evaluated 2,649 patients were in that database. That patients representing the Hungarian cancer population, because we had lots of colon cancer, breast cancer, prostate cancer, and less number in leukemia or whatever. And when the question was, what are the critical parameters to maximize the efficacy of the DDW? And we have three conclusions. The patients should survive at least 3-4 months at the start of DDW consumption. So those who are in a late stage full with cancer, we cannot guarantee that we can show real efficacy. The other was the patients should drink DDW at least for 3-4 months. It's obvious. So within 2 weeks, we cannot get a real breakthrough. And the other important was the patients should start DDW within 9 months after the diagnosis.
Yeah. So you want to be consuming it long enough, early enough in the disease.
Of course. Yeah. They are nice. So in that case, for that population, the median survival time was 11.6 year. 11.6 year. Comparing to the Hungarian population is 2.4. And the conclusion of that paper was integrating DW into the existing therapy, we can get 80% decrease in the cancer mortality. This is what we can offer. Can you imagine?
Yeah. So, I mean, basically what you're saying is this is a big effect. If this is true and if this replicates, this is a big effect. And it's not, it's an adjuvant therapy. You're adding it to the existing therapy, and it's just water, right? So you have to get DDW, deuterium depleted water, but you're just splitting the population into two groups and giving one regular water and one deuterium depleted water. It's pretty straightforward to do that. If the effects are as big as you're saying, and this is as powerful as the results you just shared indicate, why aren't I'm not aware that people are doing experiments all over the place, clinical trials in the US and everywhere else, looking at deuterium depleted water. Why is that?
It's a big question. The difficulties for us is the funding. So we are planning new clinical trials. You need 5 to 10 million euros to start a new clinical trial. And we are lack of that money. So, and those who can do that first, they will control the market, I would say. And to change the paradigm never easy. And maybe the interest. So I do not believe that the pharma industry is against us. I suppose that they have not recognized the potential of deuterium depletion. If they recognize it, then the world will solve all the problems, the metabolic disease and the cancer problems, because they will put the money, and people and the deuterium depletion will be part of the therapy. And because it's so obvious. But to convince the pharma industry that they are making some kind of drug development, I just read a paper that GSK bought a company for $10.6 billion just now, because they will have a new cancer drug for lung cancer. Then I cite in my paper that we can get a 7-fold increase for lung cancer population integrating DW into the therapy. So the only way that we can proceed if we generating more data, if we involving more scientists, and if we publish more scientific papers.
Yeah.
And one day, when the business recognize the potential, then the money will come. We will run several clinical trials, and we will integrate DDW into the therapy.
Yeah. Well, I would imagine, I mean, deuterium depleted water, you've got to make it by basically boiling water and doing a lot of steam generation. But, you know, as opposed to a novel drug, I'm guessing nobody can own a patent to deuterium depleted water or something like that. Or
No, we had a very strong position 30 years ago. Yeah, we have a patent worldwide for deuterium depleted water indicating for the cancer therapy. But the point is that we talk about deuterium depleted water, but there are very strict protocols what we recommend. What kind of ppm, what to change, how long drink, how to combining with radiotherapy, chemotherapy, hormone therapy. So if we really want to maximize the potential of DW, we need to integrate, we need to educate the patients and the oncologists. And so this is the way to move forward. If the people can buy it on the drugstore or in the supermarket, they will lose the potential of DW because we need to know how long, what type of concentration, when to stop, when to start again. So this is the reason we want to get a registered drug, because then the insurance company should cover the cost. They should spare billions of dollars because the people can go back work and paying the tax and all the money. So simple water, but on the other hand, it's require very strict rules to follow.
Yeah. So you're saying one thing you're saying, it sounds like, is you can't just sort of drink deuterium depleted water whenever you want. You have to drink a certain concentration at a certain rate, and you have to control your intake of other water too, because that will dilute the level of deuterium that you're consuming on a day-by-day basis. The research you've cited and the stuff you've observed is using a strict protocol of a known concentration of deuterium depleted water given, you know, it's a specific protocol. If you've seen, you know, direct effects on like the cell cycle and stuff at different concentrations of deuterium, so the concentration seems to matter. Did you also say that you do own a patent on something like a protocol?
Used to be had. No, we don't. Yeah, it's run out. But do you know that I wrote the book, my second book? It is deuterium depletion. So in that book, I wrote the protocols. Can I show you?
Yeah, sure. Yeah.
This is the book. People can get it on the Amazon in German also. So what I try to do to sharing the knowledge, those who can get access to DW, to see how to use it, when to use it, and how to combine it. And we need to work a lot in the future to move forward, because it is also very important. So integrating DW, it will not mean that we will cure everybody. So we don't like the pancreatic cancer, we don't like the triple negative breast cancer. We need lots of more research to figure out why we can get good response for lung cancer, but not so good response for triple negative breast cancer.
I see. So you've done experiments with different cancers. You see positive results in some cases, such as the colon cancer example we talked about, but you don't see positive results for things like triple negative breast cancer.
It's more aggressive or not so sensitive for deuterium depletion, and of course there are genetic background or some kind of reason there is behind that.
I see. And so you can drink deuterium depleted water that a human being has created so that it has less deuterium than the other water in the environment. But you also mentioned that our own metabolism can create different levels of metabolic water in our cells. So carbohydrate versus fat metabolism, for example. We talked a little bit about how oxidizing fats, takes more steps, so it results in more deuterium depletion. So the metabolic water will have less deuterium in it compared to carbohydrate oxidation. Can you say a little bit more about that with respect to diet? So a lot of people, it's becoming more and more common for people to talk about and study things like ketogenic diets, which are high in fat, low in carbohydrate. What do you sort of think about that general realm of science with respect to how it's going to affect metabolic water and deuterium levels?
I would say that, so we always recommend people keep a ketogenic type diet when drinking DW. And you know that the pyramid, which was said at the 70s, that turned it upside down. So
the food
today,
pyramid, yeah.
They changed it. So we are going back to the real world where more covering the calorie intake with more fat. That way we can reduce the average deconcentration of the metabolic water, and that way the average deconcentration of the patient. I have data for 142 ppm, for example, those who are on a ketogenic diet, and 148 or 150 ppm who are eating lots of sugar and carbohydrate. So being on a ketogenic diet can cause a 6 to 8 ppm decrease in the deconcentration of the body.
And how do you measure that? Important. How do you actually measure that in people?
Yeah, I figured out a new small device that we precipitate the exhaled water, and we collect it, and we can measure that deconcentration of that water. We publish the paper how it's changing and how we can follow day by day the changes in the deconcentration depending on what type of people. But in a human clinical study we made, in that case we measured the D level of the blood, and at the beginning it was 148, 150 ppm, and after 3 months it was 133 ppm. They consume 105 ppm, and so that way we just bring into the optimal range of the deconcentration for the insulin signal system, because 133 ppm is the best for the insulin, and this is the reason that we could reduce the insulin resistance. So we highly recommend people stop eating sugar, reduce the carbohydrate intake, and increase the fat in the diet. That in general they can cause a couple of ppm decrease of the average D level of the body, and each ppm is count. Each ppm can be very important.
Yeah, so you think, based on the experiments that you described earlier, you would say that even a small change in the ppm and the concentration of deuterium, the level of deuterium depletion of the water, that can make a difference, because you've seen examples in cell culture where the cells are sensitive to that, and you've done work in humans where presumably, remind us, what were the DDW concentrations in the water that you gave to cancer patients? Was it around this magnitude of change?
We have 6 different deconcentration: 125, 105, 85, 65, 45, and the lowest is 25 ppm. And the patients, depending on the cancer type, the staging, we recommend starting with 105 or 85, and we recommend it to change it and going down after 2-3 months, just to always challenging the cancer cells. They need to adjust everything to the lowering D level, and this is what they cannot do, hopefully.
Yeah, and so in those patients where you gave them deuterium depleted water at, say, 105 ppm, did you also measure the deuterium levels from the exhaled water throughout that intervention to see how much that actually shifted their deuterium levels in their body?
So sometimes we receive samples, even those who are buying our products and sending back samples, and we can measure it. But the point is, if they drink it, the D level will decrease it. So the kinetic we have tested in my study in dogs and cats, and then we tested lots of people. So it's not so it's good for them if they check it, whether it's lowering, but the key issue is they should be very strict, drinking only DDW. Those who are combining with beer and wine and Coca-Cola, we never can get the good results.
I see. I see. What types of experiments and what types of clinical work are you working on or hoping to work on right now or in the near future? Can you talk to us a little bit about not just your own work, but anyone else that's doing work that relates to deuterium depleted water?
Our company, that HYD LLC, is one of the company in a consortium in Europe. The University from Würzburg, the Lisbon University, and Oxford is part of that consortium, and we received the fund from the EU to check the effect of deuterium for the central nervous system. So we used a mice study, and we used the 6-oxygen dopamine. We treated half part of the brain of the mice, and the question was, if they drinking DDW with 25 ppm or normal, whether we can protect the nervous system after checking and treating with dopamine. This is a Parkinson disease model system, and it works extremely good. So we could see that the movement and all the parameters what he could follow, it was much better than the mice drinking DW. And then checking the brain, we found that we protected the area of the brain with the 25 ppm. So we are pretty sure that, and just again, going back to the mitochondria, if the mitochondria working properly, using a lower D level, the ATP is working on a better way, synthesizing more ATP, then the whole central nervous system will be more healthy and can tolerate that type of therapy or treatment. So this is running. I hope that from Oxford the study will come and they will publish it. Meantime, our difficulties, right? So I'm working on to get fund, and we are targeting investors. We are trying to get the fund to cover the cost of the next clinical trial, because okay, we are selling the products, we are happy, and it's getting stronger and more people get access, but without registering as a drug, that will not be part of the whole society, and the tendency will not change. Just I took part in a European Cancer Society conference in Hungary, and the number means today there are 20 million new cancer cases, and we lost 9.8 million, and within 25 years that will be double. So this is the prognosis for the till 2050. If we want to do that, we need to follow what we have done. If we want to turn it back and change the future, then we need to integrate DW into the therapy.
Yeah. So the results you've seen so far that you talked about earlier with regards to cancer in humans were pretty big from what I heard. And so that was a relatively small phase 2 trial. If those results replicate and they translate and you were to see them at a larger scale, that would be a major result. That would be very big news. The problem for you and anyone interested in this subject is receiving the funding to scale up and do a trial of that size, because we're talking about many millions of dollars.
Yeah. So far, do you know, it's to register a drug, it takes about $1 to $2 billion. We started with $1,500. This is how we started, so 33 years ago. And the reason that we are here, that we generated the revenue, and that revenue was used up to do research and to proceed. And this is how we could survive the last 30 years. Once we had a big investor with 1.3 million euros, in that case, we set up a facility to be able to produce DW according to the GMP rules. So we are the only company who are ready to produce DDW for clinical trials. And on the other hand, we have all the documentation which is necessary to go and apply for ethical approval and to start a clinical study. So the missing part is the funding.
Are there any final thoughts you want to leave people with, or anything you want to reiterate about deuterium depleted water or any of the biology that we discussed today?
So there are, if you check the literature, you can see that there are papers that DW was good in a long-term memory in a rat study. We have published a paper with Vanderbilt University, the anti-aging effect of deuterium-depleted water with a Cellular Habitus elegans system. So what I try to say that I never wanted to find a cancer drug. I wanted to find the mechanism how the life is organized. And that deuterium hydrogen ratio is a key mechanism. So all the process, all the disease, some way is coupled with the deuterium hydrogen ratio. And I guess we will discover lots of new things in the future. And I hope that the scientists, more and more scientists will be involved and more data will come.
All right. Well, I want to thank you again for your time, Gábor, especially joining me from all the way in Hungary with the time zone difference. Really appreciate it. Thank you very much. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Ketosis formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.